Welcome back. My name is Luis Montaner. I'm at the Wistar Institute, and I'm really pleased to welcome you to the session on HIV reservoirs and persistence, back to basics. Please note that the chat is active, and we invite you to put in your questions during the discussion is played, so that you can, the presentations are played, so that you can participate in the discussion, and we can pick up those questions at the end as well. Please also note that there's going to be a discussion session at the end of all the presentations, so please stay for that as well. So we're going to hear from multiple speakers on this session. Mr. Will Carter will address the community perspective as the BEAT-HIV Community Advisory Board Chair. We will hear from Dr. Siliciano on the HIV reservoir dynamics on Hopkins University, Dr. Su Yu from Harvard University on the reservoir profiles and elite controllers, Dr. Chahroudi from Emory University on new approaches to reactivate the reservoir, from Mike Betts here from UPenn on a novel genetic strategy to identify HIV-infected CD4 T cells, and from Dr. Uri Mbonye, sorry if I can pronounce it correctly, from Case Western CFAR, as an ESI abstract awardee. So hope you enjoyed the discussions, and we'll see you at the question and session. Thanks.
I got involved in HIV/AIDS research sort of on a whim from taking a class down at Philadelphia Fight called Project Teach Outside. Well, I learned about the cures were going on, and I was asked, would I get involved? And I quickly said no. And then I was invited to a community advisory board to see if I would be interested in joining, which I end up doing, and which is called the BEAT-HIV for the Cure, part of the Martin Delaney Collaboratory. And now, 2 years later, I am chair of the group. And as a HIV and AIDS activist, I had decided that the trial was not for me to do. There was other work to do. But then my sister, who I look up to, my little big sister, called me a hypocrite. She says, how dare you ask other people to interrupt their medicine when they're doing fine, and you don't even know if you're qualified for it or not. How could you do that? And how can you sit there and make decisions on other people's lives when you're not even doing it on your own? So I got my arms twisted and opened up my eyes to it. So that's how I got involved. My experience was, I had a great experience with this one particular trial that I'm on. I'm actually still currently doing a trial right now, but the one that interrupted my medicine, I went almost, I want to say 13 months, maybe 14 months without taking any HIV meds whatsoever. I was ecstatic about it. I felt good. I was given all the side effects that could happen, and I experienced one. And that was on the very first day of the trial for me, where I just experienced the chills. It was a chill that I never had before, to be honest. It went all the way to my bone, and the people at the center took care of me. You know, I had a couple of blankets. So I'm anemic anyway, so I'm making excuses. That was part of the reason for it. But the whole entire experience was freeing. It was liberating. When I did this trial, I became a trailblazer. I thought of people who went before me, who have done this, people who have taken hundreds and hundreds of pills a day. And I'm just taking one pill, but to think that I can go once a year to the doctor and don't have to worry about it again, I'm like, wow. And then there was a time during the trial I wanted to back out, almost from the beginning, but that was when Timothy Ray Brown had passed. And knowing that he did not die from AIDS or HIV related things, you know, once again, my sister stepped in and called me a hypocrite. She said, like, how dare you? You're asking other people to do this. You better think about what you're doing. But I am so glad that I did this. I met a bunch of fantastic people. Some of the work by being chair that I did, I got to see take place. I got to see the staff who weren't no longer a mystery to me, wondering what they look like, tearing down stereotypes of different scientists and researchers who are mystery to the masses of people. We don't really communicate because they think they don't know how to talk to us, and vice versa. So it's a mutual thing, but being there, and then each and every week that I would go to see if I was okay and just to monitor me, I felt presidential. I tell myself I went to the Walter Reed Center every week. I had my own personal team who contacted my private doctors, who were very well pleased with the way, you know, things were going. I had a good run with it. Unfortunately, right now I am detectable, but if I'm working towards becoming undetectable. Again, my numbers are improving each and every month. So I don't feel like I was bamboozled, mistreated, or anything like that at all, because I can say, at least for the researchers here, they actually cared. And the head nurse researcher that was over the, I seen him every week. We went over that consent form, even though I know it because I helped write it and were participated. I wasn't the writer, but I did have my little word in there. But the fact that he would ask and explain everything to me as if he didn't know that I already knew this. But so I knew each and every week why I was there, why I wanted to be there. And I'm going to say, for me personally, it helped save me emotionally, mentally, and I did suffer some depression through it when I had to, even though I completed the trial, I went into an extension part of it, but that's when my numbers had dropped, so which made me detectable. So I did suffer a little depression, but to have the staff reach out to me and still reach out to me to find out if I'm okay and if I need any help, and for them to contact my doctors and still work together, it was a fantastic, fantastic experience. If I could do it again, I would. Last night I was at a meeting where someone mentioned that maybe, you know, taking a few months off and becoming undetectable again and seeing how would that affect. So it made me want to think, so what if they were to do this for those who, you know, become detectable and then may get the numbers under control again? How would that affect? So it calls you to think, and it makes me tell each of the researchers and the scientists, don't stop dreaming, don't stop reaching for the stars, because if you do that, you kill all of my dreams, my ambitions that I now have and I thrive to have. I wish to see my youngest granddaughter graduate college and get married. So keep talking with your community advisory boards and get involved. You have to, so don't stop dreaming. Just don't. First, I would tell them to pray over the matter. I had a very strong support group. One, my sister, I'm going to mention her name, Constance Carter. Having her in my ear and relying on the community groups, which I do go to and support different groups, speaking with them. And these are also my peers who are also living with HIV. That's not for, it may not be for everyone to participate in that trial. But I tell them, if you start the conversation, number one, it opens the door for people to listen. It tears down walls and opens doors. Number two, the more you know, the more you can grow. And people, not saying they aren't dying from complications due to it, but they're not as frequently as they used to be. And cure research opens doors. You know, you have television shows now about it. If it just went off the air, the show Pose took me back to when I was in my 20s and in my 30s, when people were dying every 3 minutes. And now, when you would see the people that were on AZT and who were on other things, interferon and all those things, to be taking over almost 200 pills a day to do one. So if you don't have somebody willing to try it, that's a problem. But there's other ways of supporting cure research if you do not decide to be a participant in it. Handing out flyers, talking to your doctors about it, getting them information to it. I felt blessed that I am a disabled person, and I'm recently walking. You know, I was in a wheelchair 14 years, in the hospital more than I was home, raising 2 kids. I actually commanded my doctor's office. I had 9 of his partners came in, and I was able, because I go to a teaching hospital, I was able to have a conversation with them because they all seen me in a wheelchair, feeling down and depressed. And now I'm an active member of society and my community, telling people about cure research. So even if you don't participate, don't shut the door and don't close of it. And things, tearing down the stigmatism, and also the myths, the way, you know, I'm a guinea pig or anything like that. It may have been that way at one time, but you can't tell me that you can't, things don't change and things are the same. I go to a very reputable place. So you need to do this. You need to investigate. Things change. And that way you have a voice. I have a famous analogy that people kind of know me for. Don't invite me into the table any longer. Invite me into the kitchen. Let me help you, the researchers, come up with how you're doing this. Don't include me after you've already had the product. Because the one thing, no matter how smart you are, or how much you may know that you feel this may work, guess what? Researchers, you can't take your own medicine. You need me as much as I need you. That's why I'm begging you to keep dreaming and having conversations, talking with your CAB members, inviting people to join the community advisory boards, inviting them to maybe come in and just listen. You know, go to different organizations, but open a conversation. So by doing that, you're involved and you are a participant. You are a hero then.
Hi, I'm Bob Siliciano from Johns Hopkins University. I'd like to discuss one aspect of HIV reservoir dynamics, and that is viral rebound. The real problem with finding a cure for HIV infection is shown on this slide. It's the very long half-life of the latent reservoir resting CD4 positive T cells. And in principle, reactivation of a single latently infected cell can give rise to rebound. Now, in recent years, we've learned that rebound is actually quite a complex process influenced by many factors, which I will go through. The first is the size of the latent reservoir. Long delays in time to viral rebound have really been only seen when there's been a dramatic reduction in reservoir size. And therefore, it's important to be able to accurately measure the size of the reservoir. The original reservoir assay was a viral outgrowth assay that was complex, and many investigators therefore resorted to simple PCR-based assays to detect integrated proviruses. The problem with that is that we've shown that many of the proviruses that persist in treated individuals have fatal defects, including large deletions and APOBEC-mediated hypermutation, and therefore single amplicon PCRs are a very poor way to measure the reservoir of intact proviruses that cause viral rebound. For this reason, we've used digital droplet PCR to develop an assay in which individual proviruses suspended in nanoliter-sized droplets are interrogated by multiple informative PCR reactions to distinguish intact and defective proviruses. And in this intact proviral DNA assay, intact proviruses can be directly counted as double positive droplets, allowing us in a very easy and scalable way to get a good idea of the size of the reservoir of intact proviruses that contribute to rebound. So reservoir size is one factor which we can now measure with this intact proviral DNA assay, but reservoir composition is also important. And it's become clear that the reservoir is actually dominated by large clones of infected cells. And this process was initially surprising because the reservoir was defined in resting CD4 positive T cells. It now appears that many of the cells in the reservoir have some history of proliferation. And so this process of clonal expansion is important to understand. Our first indication that clonal expansion was occurring came from the analysis of residual viremia, the trace level of free virus in the plasma of treated individuals, which we thought could represent the activation of latently infected cells that produce virus that can't spread due to the drugs, but which could be detected with a sensitive enough assay. And sequence analysis of this residual viremia revealed the enormous diversification that occurs following initial infection, but also showed that the residual viremia in many individuals was dominated by sets of identical sequences. Many independent limiting dilution PCR reactions giving precisely the same sequence. And this, we thought, must reflect the proliferation of infected cells that would copy the viral genome without error into progeny cells. So the cells that we need to eliminate to achieve a cure can actually proliferate in vivo. And this concept was confirmed by integration site analysis done by other groups, which also suggested that the site of integration may actually contribute in some cases to this proliferative process. We've explored a different hypothesis for clonal expansion, namely that it's actually driven by normal responses to antigen. And Francesco Simonetti in our group has isolated antigen-reactive CD4 positive T cells from infected individuals by sorting for cells that upregulate activation markers following stimulation with antigen. He also obtains the cells that don't respond as a negative control and cells that respond non-specifically to CD3, CD28. These are then plated in small pools and we identify cells that contain a provirus, a sequence to find proviruses of interest, and then carry out a variety of assays that give us a picture of this clonal selection, clonal expansion process. So we found that among the CMV, among cells responding to CMV as an antigen, there were large clones of infected cells that had exactly the same integrated provirus, the same integration site. And some of these clones comprised over 80% of the HIV-infected CMV responsive cells. Some clones were large enough to be detected even among cells responding non-specifically to CD3, CD28, but were not seen among the cells that did not respond to CMV, confirming the specificity of this process. And Francesco has been able to determine 4 critical properties of individual antigen-responsive cells in this system. The antigen, the full proviral sequence, whether it's intact or defective or not, the integration site, and the T-cell receptor beta chain sequence. And this has led to a lot of interesting findings. To me, the most interesting of which is the timing of proliferation relative to infection. In some cases, it appears that CD4 positive T cell will respond to antigen, proliferate, and then one member of the clone will become infected, and thereafter both infected and uninfected members of the clone will continue to proliferate. In other cases, essentially all of the antigen-specific cells carry the same provirus, indicating that infection occurred very early on in the response to antigen. And we've seen both of these patterns in very large clones that are too large to be explained by homeostatic proliferation. And some of these clones contain integration sites previously associated with proliferative processes, but many of them don't, indicating that antigen is probably the key factor driving this clonal expansion. So antigen can drive clonal expansion, but interestingly, can also reverse latency, leading to virus production, and likely in many cases to death of the infected cell. This raises the issue of proviral inducibility, that is whether antigen-driven proliferation is always directly coupled with a reversal of latency. And we think that in many cases there may be some uncoupling, which we are trying to understand right now. Now, a final point that I want to make about this clonal expansion process has to do with the fact, an observation really made by Katie Barr and Michelle Nussensweig, that in many cases, very large clones detected in the latent reservoir using the viral outgrowth assay don't actually appear to contribute to rebound. So viruses rebounding after treatment interruption are often genetically distinct from the large clones detected in the viral outgrowth assay, even though these large clones are clearly replication competent and inducible. So here's one dramatic example. And there have been a number of explanations proposed for this discrepancy shown here, but we have advanced a very simple alternative hypothesis, namely that autologous neutralizing antibodies block outgrowth of many viruses in the reservoir. In the past, it's been difficult to determine factors that affect in vitro outgrowth of replication competent viruses because the frequency of infected cells is low and the number of isolates obtained in most of these experiments is low. So we've used leukapheresis to obtain large numbers of resting CD4 cells and plated them either in the absence of antibody with control antibodies from HIV negative donors or with autologous IgG purified from leukapheresis plasma, and then simply looked to see whether autologous IgG causes a reduction in outgrowth, and compared to cultures without antibody or with control antibodies, cultures with autologous IgG cause a dramatic reduction in outgrowth in some donors. And this effect is likely an underestimate of the in vivo effect because the concentration of antibodies used in these experiments is way less than the normal concentration of IgG in circulation. So on average, we get a 4.6-fold reduction in IUPM by including autologous IgG. And in 6 of 15 donors studied, more than 80% of the viruses in the reservoir had outgrowth that was blocked by autologous IgG. So to understand this process, we've sequenced viruses in the reservoir and carried out neutralization assays. The best sort of picture of the reservoirs obtained by looking at first at viruses that grow out in the absence of autologous antibodies. You can see in this donor, there's a diversity of viruses, but one large clone of identical viruses. Now, most of the viruses that grow out in the presence of autologous IgG are located in the upper branch of this tree, which has sequence differences in the variable regions in GP120. And in pseudovirus neutralization assays, most of the viruses in this upper branch are highly resistant, as indicated by the red colors, resistant to neutralization by autologous IgG. But the tree also contains viruses that are highly sensitive to neutralization, including this large clone. So the presence of large antibody-sensitive clones provides a simple explanation for the discrepancy between outgrowth and rebound. And in additional studies, we've established a phylogenetic relationship between the viruses that grow out in the presence of autologous IgG and the viruses that cause rebound in vivo. So this is important because this is actually a form of post-treatment control that is occurring essentially in everybody with HIV infection. It's just that it's control of only some of the viruses in the reservoir. So rebound really involves a selection process in which some of the viruses in the reservoir are already neutralized by autologous antibodies. And rebound involves a selection for those viruses that are not neutralized or that are resistant to autologous antibodies. So we find it an optimistic finding that at least a substantial a fraction of the reservoir viruses are neutralized, and that only a minority of viruses can actually cause rebound. I should point out that in some cases, the large expanded clones are antibody resistant. Nevertheless, it's important to realize that once a patient starts an antiretroviral therapy, further evolution of the virus and the evolution of resistance is blocked. And so what we have to do is find a way to induce an antibody response to the subset of reservoir viruses that are resistant to autologous neutralization. So I'll stop there. Thank the people involved in this work, including my wife, Janet, who's helped me with these studies for many years. Francesco Simonetti, who did all of the work on clonal expansion that I showed, and Joe Variali and Lynn Bertagnoli, who did the work on autologous neutralizing antibodies. I'd also like to thank many collaborators at different institutions, the funding agencies, and Hopkins Center for AIDS Research. Thank you.
Many thanks to the organizers for the invitation. In today's presentation, I will quickly review what we learned from HIV/ED controllers. HIV/ED controller represents a very small proportion of all HIV-infected individuals, but they are probably the best model for studying a HIV cure. There are two questions that we can address using this cohort. Can a sterilizing cure of HIV infection occur during natural infection? And what are the characteristics of a functional cure of HIV infection? To address these questions, we studied 73 ED controllers and a reference cohort of 41 ART-suppressed HIV-infected individuals. Both cohorts had a median of 9 years of recorded undetectable viremia, but ED controllers did not receive any antiretroviral therapy. We first analyzed the proviral reservoirs in these two study cohorts using near full-length proviral sequencing assays, which allow us to distinguish intact, potentially replication-competent proviruses from the majority of the proviruses that are defective. We find that there are two HIV exceptional ED controllers who had no detectable intact proviruses despite analyzing large amounts of cells. The question here is whether a sterilizing cure of HIV infection may have occurred in our exceptional ED controllers. As you know, there were only 2 cases of a sterilizing cure of HIV that have been reported thus far, the Berlin patient and the Linden patient. Both of them received stem cell transplants from donors with homozygous CCR5 delta 32 mutations. Extensive virologic testing was done in the Berlin patient. Despite analyzing 1.4 billion CD4 T cells in viral outgrowth assays, no replication-competent viruses were detected. A more limited number of cells were studied in the Linden patient. It seems that a small amount of HIV DNA was detected in memory CD4 T cells and lymph node cells from the Linden patient, as well as in the rectal biopsies from the Berlin patient. However, it was not clear if these proviruses represent genome intact or defective viral DNA. In our first exceptional ED controller, we failed to detect intact or replication-competent proviruses despite analyzing more than 1.5 billion peripheral blood cells using full genome sequencing assays and 340 million resting CD4 T cells in viral outgrowth assays. The second exceptional ED controller, named the Esperanza patient, had no sign of intact or replication-competent proviruses after analyzing 1.19 billion peripheral blood cells, 503 million mononuclear cells from the placenta, and 150 million resting CD4 T cells using different assays. In both patients, we detected multiple defective proviruses with hypermutations or large deletions, which clearly indicate that the patients were infected with HIV previously, and that active cycles of viral replication had occurred in the past in these patients. Our data indicate that similarly to the Berlin patient, the two exceptional ED controllers had no detectable intact HIV DNA in massive number of cells that were analyzed using complementary assays. Although it cannot be definitively proven, it is likely that these two exceptional ED controllers have achieved a sterilizing cure of HIV through their natural immunity. Now I'm going to switch gears to look into the majority of the HIV ED controllers in our study, who can maintain undetectable levels of HIV replication for decades, despite very clearly detectable intact proviral reservoirs. Using a novel next-generation sequencing assay, we were able to profile both near full-length proviral sequences as well as their corresponding chromosome integration sites for individual proviruses. Here's an example of integration site profile of intact proviruses in a relatively long-term ART-treated individual. As you can see on this phylogenetic tree, the intact proviruses are quite diverse in this patient, and 90% of the intact proviruses are integrated into genic regions, and about 10% in non-genic or pseudogenic regions. In contrast, here's an example of an HIV ED controller who had no detectable viremia, at least for the past 12 years. The proviral sequencing analysis showed that there are two large clusters of clonal expanded intact proviruses in this ED controller. One intact proviral clone is integrated into centromeric satellite DNA region, also referred to as a gene desert. The second clone of intact proviruses is integrated into a CRAB CF gene in chromosome 19, which is a heterochromatin region with a significant enrichment of repressive chromatin marks. Intact proviruses integrated into both of these regions are likely in a state of deep viral latency. Overall, we found that over 65% of intact proviruses in ED controllers were integrated into non-genic regions, satellite DNA regions, or genic heterochromatin regions, all of which are associated with deep latency. However, the integration site of defective proviruses in ED controllers resembles what we see in ART-suppressed individuals, with the majority located in regular genic regions. This indicates that proviral integration by itself is not biased towards non-genic or heterochromatin regions in ED controllers. Instead, the integration site profile of intact proviruses in ED controllers is likely the result of immune selection pressure that was able to effectively eliminate intact proviruses in more permissive regions. These data indicate that location matters. When the intact provirus is integrated into open chromatin, it is more likely to be reactivated. That's also more likely to be recognized by the immune system. However, if an intact provirus is integrated into non-genic or heterochromatin regions, it is likely in a deeper state of latency and protected from immune recognition. We refer to this state as a blocked and locked state. Here's what we propose is happening in these ED controllers. Intact proviruses in open chromatin are gradually eliminated over time, while intact proviruses in deeper latency persist and even expand. Eventually, the remaining intact reservoir may be largely confined to heterochromatin positions where HIV is blocked and locked, and this reservoir profile enables a drug-free remission or a functional cure of HIV infection in these ED controllers. Similarly, in the exceptional ED controllers, all intact proviral reservoirs were completely cleared by the person's immune system, and the patients may then approach a sterilizing cure of HIV infection. To quickly summarize, a sterilizing cure of HIV may represent a rare but possible outcome of HIV infection once all replication competent or genome intact proviruses are eliminated through natural immunity. Long-term remission or functional cure of HIV infection could possibly be achieved in an HIV-infected individual once all intact proviruses are in the blocked and locked positions in the host genome, similar to the reservoir profile seen in HIV ED controllers. These data suggest that not the frequency of intact proviruses by itself but instead their chromosome locations may be an important distinguishing feature for a functional cure of HIV infection. Finally, I would like to thank all group members in my lab and the Lichtenfeld lab, and all our national and international collaborators. We're also extremely grateful to all our study participants, especially Ms. Lauren Willenberg and the Esperanza patient. Their generosity make these studies possible. And thank you for your attention.
Good afternoon, everyone, and thank you to the Penn CFAR for inviting me to give this short talk to you today. I'm excited to tell you about some work we've been conducting on novel approaches to reactivate the HIV and SIV reservoir. The main barrier to an HIV cure is the latent HIV reservoir that has been described to exist in resting memory CD4 T cells. Over the years, a number of other cell subsets within that category of resting memory CD4 T cells has been characterized in terms of their susceptibility to HIV infection, their capacity for self-renewal and long-term survival, their various mechanisms of proliferation. And together, these all represent a category of cells that we are looking to target when we are testing HIV cure strategies. So what I'll describe to you today in these HIV-SIV reactivation approaches are part of the shock and kill strategy for HIV cure, or kick and kill strategy is another name for it. And what I'll be focusing on primarily is this first half of that strategy, which is the latency reversal. And that's depicted here on this cartoon, where integrated HIV DNA is present within the nucleus of a resting CD4 T cell. And that cell, or that non-human primate, or that person is treated with a latency reversing agent that causes activation of that cell and transcription of the HIV DNA into RNA with protein production, new virion production. This is all done under the context of antiretroviral therapy, such that those new HIV virions cannot go on to infect new cells. But what this process allows for, or is targeted to allow for, is recognition of that latency, that cell with reverse latency, by the immune system for elimination. So there have been a number of latency reversing approaches that have been tried in vitro, ex vivo, in vivo in various preclinical models, as well as in people. And there have been some modest successes in the clinical trials that have been conducted so far. A lot of these are what we call LRA 1.0, which have been testing compounds that target gene expression that is not specific to CD4 T cells, such as epigenetic modifiers. LRA 2.0, what I'm referring to here, is the focus on strategies aimed at targeting CD4 T cell specific pathways, leading to cellular transcription factor activation, and as well as HIV expression. And this is just sort of a nice cartoon that shows a number of the different targets of these LRA 2.0 approaches and what their effects are on intracellular signaling in CD4 T cells. Today, I will focus on work we have done in collaboration with a number of other groups targeting the non-canonical NF-kappa B pathway for stimulation. In addition, I will talk about some work we've published on a combination approach using an IL-15 superagonist with CD8 depletion, or elimination of CD8 cells temporarily in non-human primates. There also has been some promising work both in humans and non-human primates on immune checkpoint blockade and TLR agonists as latency reversing approaches. But I don't have time to go into those today, and they have been there have been some conflicting reports as well as to their efficacy. So in general, when we are testing these latency reversing agents in preclinical models, and specifically I'll be talking about non-human primate models, we have a design of the experiment that you can see depicted here, and this is just one example. And there are a number of different parameters that we can manipulate during the course of these experiments, including which virus we use, how much of that virus we use, what's the route of infection, when do we start antiretroviral therapy, how long do we treat with antiretroviral therapy before we try latency reversal, et cetera. But the main point I want to make here is that in most of these studies that I'll review today, we have infected animals started antiretroviral therapy after several weeks of infection, and then after suppression of viremia in peripheral blood below the limit of detection of our commonly used assay, which is 60 copies per ml, we then utilize a latency reversing agent. And when we talk about latency reversal in this non-human primate model, primarily we're talking about the ability to detect SIV RNA in the plasma of these animals when they get treated with the latency reversing agent while they continue to receive daily antiretroviral therapy. And that detection of SIV RNA in the plasma is above the limit of detection of that standard plasma viral load assay, or 60 copies, as I mentioned before. So the first study I will take you through today was published in 2020 in Nature, and it was a collaboration between the CARE Martin Delaney Collaboratory and the Emory Consortium for Innovative AIDS Research in Non-Human Primates. The NHP work was led by Mon Mavignier, a former postdoc in the lab who is now an assistant professor at Emory. And we took advantage of some existing data that suggested that targeting the non-canonical NF-kappa B signaling pathway might be an effective way to reverse HIV latency in vitro and ex vivo. And that non-canonical NF-kappa B pathway is shown here, where SMAC, or the second mitochondrial activator of caspases, is shown to be blocking the activity of CIAP. This is also known as a CIAP antagonist. When a SMAC mimetic or a CIAP antagonist is utilized, it then allows for progression of downstream events that then lead to translocation of the active dimer for non-canonical NF-kappa B signaling to the nucleus and transcription of host genes. And the RNA sequencing data from that study is shown on the right side of the slide, where we're just to highlight the fact that we were able to show upregulation following treatment of non-human primates with a SMAC mimetic of specific non-canonical NF-kappa B signaling genes. But what I most wanted to show you here is the effect on the plasma viral loads. And so on the top half of this slide, you can see the viral loads in the animals after initial infection and during the period of ART suppression of viremia, which is indicated by the gray shading. The second half of the slide, the bottom half of the slide, remember we're continuing antiretroviral therapy throughout the study, but on the left, we're giving 10 doses of the CIAP antagonist or SMAC mimetic AZD5582. And you can see what I was referring to before as the induction of SIV RNA above the limit of detection of 60 copies per ml. This occurred in 5 out of 9 animals who received 10 doses of AZD5582. And the corresponding animals who received placebo showed no evidence of virus production. The second study I'll take you through briefly here is another way of reversing latency that we showed in non-human primates. And this study was led by a former graduate student of Guido Silvestri's, Julia McBrien. Now, this study was a combination of 2 different agents. So the first was N803, which is an IL-15 superagonist, and that had some pre-existing evidence of perhaps showing HIV induction in ex vivo and in vitro, which is why it was utilized in this non-human primate study. And it was utilized in addition to MT807R1, which is a molecule, an antibody that can deplete CD8 alpha expressing cells, which include CD8 T cells and NK cells. So this experimental design was similar to what I showed you before, where animals are infected with SIV, treated with antiretroviral therapy for about a year, and then exposed to the IL-15 superagonist alone, CD8 depletion alone, or the combination. And what I'm showing you on the right side of the slide is the RNA sequencing results of comparing pre and post treatment in these 3 different groups. And the main thing that I wanted to highlight is that in the N803 group, we're seeing activation of signaling pathways involved in proliferation, activation, antiviral responses, and other intracellular signaling pathways. And they're not very different or divergent between N803 alone and when N803 is combined with CD8 depletion, suggesting that CD4 T cells in both these cases are responding similarly to that N803 stimulation, and quite different from when CD8 depletion was used alone. So now getting to the virologic effects of that study. On the top, I'm showing you a graph of all of the groups and their period of viremia before antiretroviral therapy was started, and then antiretroviral or suppression of viremia by antiretroviral therapy. On the bottom, I'm just showing you the N803 alone group and then the combination of N803 with CD8 depletion. And what you can clearly see is that when this combination approach is used, there is induction of HIV production and induction of SIV production and SIV viremia above this black line, which is drawn at 60 copies per ml in 14 out of 14 animals here. Whereas on the left, when N803 is used alone, we found it to be an ineffective latency reversing agent in vivo. So I've shown you the latency reversal effect that we found when we used the CIAP antagonist or SMAC mimetic AZD5582 alone in non-human primates. And I've also shown you the latency reversal effect of the combination of the IL-15 superagonist with CD8 depletion. So next, we wanted to take a look at what would happen if we combined CD8 depletion with the CIAP antagonist AZD5582. And this was another study that was led by Mbonye, and it involves 6 rhesus macaques that were infected with SIV, treated with antiretroviral therapy for a long period of time prior to the intervention phase. That included the CD8 alpha depleting antibody and 5 doses of AZD5582. And we found that 6 out of 6 animals had episodes of on-art viremia above 60 copies per ml of blood during that treatment period. And we're comparing here on the right, these animals in the combination group to the animals I described before in the AZD5582 only group. And we're only focusing on the first 5 doses to keep the comparisons fair. And I think what you can see is that in each case, we saw an elevation from baseline of the plasma SIV RNA during the treatment phase. But in this case, we had 6 out of 6 that all had elevations, whereas here there were some animals that were more stable during that intervention phase. And in some unpublished work where we're investigating additional combinations of latency reversing agents, we've now conducted a study where we are giving both AZD5582 and N803 together. There's no CD8 depletion involved in this study. And this work is led by Amir Dashti, who is a former postdoc in the lab as well. And what you can see in animals that receive antiretroviral therapy alone, we only have 2 incidences where one animal went above 60 copies per ml, which is indicated by this black line here. And when animals received AZD5582 alone, we've now confirmed our prior findings. So this is 7 out of 9 animals have evidence of latency reversal or on-art viremia. And then when we combine AZD5582 with N803, we see a similar pattern, but in this case, 9 out of 9 or 100% of our animals show evidence of on-art viremia. This is actually a study where we are giving an additional clearance agent, so there's a lot more data to come on kind of testing that full shock and kill approach. So in summary, I hope I've convinced you that targeting the signaling pathways in CD4 T cells that activate cellular transcription factors to promote virus production is a promising approach to reverse latency. We think that combination latency reversal approaches, such as I've described with N803 and CD8 depletion, AZD5582 and CD8 depletion, and then the combination of AZD5582 with N803, may in fact be more effective at enhancing virus reactivation. And there are a number of additional pathways that have been explored and combinations that have been explored ex vivo in patient-derived cells. And I think each of those, they're very intriguing data, but each of those should be tested in preclinical models for safety and efficacy. In addition, as I alluded to earlier, identifying clearance agents that we can combine with these latency reversing agents to actually eliminate infected cells following their latency reversal will be a crucial step to bring us closer to an HIV cure. So thank you for your attention. I'd like to acknowledge the folks in my group who contributed to this work, primarily Maud Mbonye and Amir Dashti, as well as Julia McBrien and Guido Silvestri's group. We have a number of excellent collaborators at the Yerkes Primate Research Center, as well as the UNC HIV Cure Center, Frederick National Labs, Los Alamos, and the Vaccine Research Center. Thanks for your attention. Thank you to the organizers for inviting me, our funding, and I'm looking forward to the panel discussion later on today.
Hi, my name is Michael Betts from the University of Pennsylvania in the Department of Microbiology. Today, I'm going to tell you about studies we've been performing in the lab to try to identify HIV-infected CD4 T cells using a novel genomic strategy. These studies were conducted by a very talented graduate student in my lab, Vincent Wu. So what are the challenges for defining the HIV reservoir? First and foremost is the rarity of infected cells. It is known that less than one CD4 T cell in 1,000 is infected with HIV in the latent phase of infection. And it's also known that the virus becomes transcriptionally silent and infected cells may not express any viral transcripts or viral proteins on their cell surface. This is compounded with the heterogeneity of the CD4 T cell population itself, which can be highly variable with different functions, different residency characteristics where they may become lodged in tissue and therefore not sampled equivalently in the blood, et cetera. And we have identified potential sub-reservoirs based on this using integration analysis, looking at lymph nodes and blood, which we previously published. Furthermore, studying the reservoir itself is complicated because how do you identify these cells? And typically what has been done is to activate these cells in vitro or to use in vitro models. And the problem with in activating T cells is that this biases the phenotype, the function, and the characteristics of those cells, and also may not give a true characteristic of what the epigenetic status of the virus is within the infected cell. And so based upon all of this, I would say that the majority of all of our knowledge of the HIV reservoir is based on inferences from in vitro models or in vitro reactivated infected primary T cells. So we wanted to determine if we could identify HIV-infected CD4 T cells directly ex vivo without requiring activation, viral RNA expression, or cell surface protein expression to allow us to really potentially understand the latent cell reservoir. So to achieve that, we hypothesized that we could use a technique called single-cell ATAC-seq, which is used to identify open chromatin. And we hypothesized that integrated HIV provirus could be detected if the virus was an open chromatin or if there were even small pieces of virus accessible in open chromatin. So to further identify what the cell was, we hypothesized that we could then use single-cell antigen select antigen profiling, a technique called SC-SAP, which uses oligo-tagged antibodies of any specificity that you like, and you can put hundreds of these together in combination to allow a high-resolution identity of the cell that you're interrogating. So the combination of these techniques is known as ASAP-seq, which is ATAC with select antigen profiling. This was a technique just recently published from Peter Smybert's lab in Nature Biotechnology. So the first part is really what I would consider the easy part, which is actually the experimental strategy. And so this is where we would take the cells of choice and put through this protocol. So I'll present today data from two different experiments that we've done, or two different strategies that we've done. First, an in vitro proof of concept system, and then an ex vivo analysis of T cells. So for the in vitro side, and here in red, really, if you want to use this, you could just use your favorite in vitro primary cell model. For what we did that I'll show you later, we just simply enriched CD4 T cells from an HIV negative individual. We activated those cells for 2 days, infected them with a CCR5 tropic lab stock of virus, and then we rested them for 2 days. For the ex vivo side of the experiments, again, you could just use your favorite source of cells. All we did was we enriched peripheral blood or lymph node memory CD4 T cells by negative selection. There was no activation of these cells at all. We then take that forward into the ASAP-seq protocol, which essentially just takes the cells shown here. We label them with the cocktail of the oligo-tagged antibodies to label the surface markers. We then take that through a single cell ATAC-seq on a 10x genomics instrument. We purify down the ATAC-derived tags, antibody-derived tags, and then we do a lot of sequencing to get reads that would be then corresponding to both the genome of the cell as well as the surface oligos that came with the antibodies through the tagging procedure. Then becomes the hard part, which is how do you then use these data to identify HIV-infected cells? And as you can see by the pie chart there, about 80% of the work of this entire protocol is really dry lab and analyzing this data and figuring out how to identify things. So going through the process, you take the raw reads, you align them, and the critical part here really is the alignment. Because we have to align, the concept is that we could align the ATAC reads to both the human genome as well as the viral genome. And we had to figure out a way to combine those things together. And so to do that, we take the known reference or the autologous viral sequence from the individual, if we used an individual cells, an HIV infected individual cells. And then we essentially do multiple ways of aligning and cleaning up the data. And then Vincent wrote his own program to align the genomes with virus to identify the infected cells on a per cell level. We then take that through the rest of the process and clean it up for visualization. So for our first proof of concept experiment, we used in vitro infected CD4 T cells from the blood of an HIV negative individual. We simply enriched CD4 T cells, activated them for 2 days, infected them with a CCR5 tropic virus, rested those cells for 2 more days, and then performed the ASAP-seq procedure. As you can see on the screen here, a UMAP projection of all the cells based on the ATAC reads that is an unbiased clustering of these cells into different populations based on open chromatin for different genes. And so we can see multiple clusters were identified, which we can then begin to annotate using the cell surface profiling that we obtained from the antigen profiling labeling on the surface. And so as you can see here, 7 of the different markers that we assessed out of the 150, and we can then annotate these clusters directly. For example, cluster C1, in fact, turns out not to be CD4 T cells, but these are contaminating B cells, as I will show you on the next slide. And so we annotate all of these, and we can find monocytes contamination, B cell contamination, but then we have all of these different CD4 T cell populations shown here on the left part of the UMAP that we can annotate into different stages of differentiation or activation. Then we can align, using the alignment from the ATAC, we can identify which of those cells actually have HIV integrated viral DNA. So on this second UMAP, every red dot is one of those cells that showed up as having integrated proviral DNA. We can quantify those precisely. And so as shown on the top right, we obtained data from around 8,000 cells total, 5,700 of which were not showing any evidence of viral integration, whereas 1,300 of them showed presence of viral DNA. And we can very precisely then map those to the clusters shown here, and we can determine exactly which cell types are the ones most likely to be infected. We can further, on a single cell basis, project the exact profile of these cells based on all of the markers that we labeled at the beginning. And I don't expect you to see on these particular plots. Suffice to say that for every single red cell there, we're able to generate a single cell plot like this to show exactly the cell surface markers that they express. We can boil all that down then as the population into looking at what are the most significant differences of the cells that are HIV infected compared to the HIV negative, for instance. And we can see the top surface markers that characterize those infected cells. And so here it's actually quite nice to see, in fact, that CCR5 was the very most strongly expressed marker that differentiated the HIV infected cells from the HIV negative cells. And this is very clear because we used a CCR5 tropic virus in the first place. So that's what we can attain with the surface profiling of the cell. And then just as an example of the epigenetic profiling, we can further go in and we can, for example, look for differential transcription factor motifs that might be expressed in the HIV positive cells versus the HIV negative cells. And for example, here we can see transcription motifs for the FOS and June pathway being highly enriched in the infected cells versus the uninfected cells. And it's notable here that this is not simply just due to activation, because in fact, on both of these plots, the HIV negative cells are also selected to be the activated cells. So we're comparing activated versus activated cells, and yet we still see that the HIV infected cells have an enrichment for differential transcription factor motifs. So this was a relatively easy bar to cross for this technique, which was an in vitro activated T cell. So the next question was, can we apply this to samples from infected individuals? And so for our first experiment, we tested a lymph node sample, which granted is perhaps a little bit of a higher bar, but we selectively chose this because this was a lymph node from an acutely infected individual who had a high viral load, and we hypothesized that we would see a lot of infected cells from that lymph node. So again, the simplicity of the wet lab technique, all we did here was enrich lymph node memory CD4 T cells by negative selection. There was no activation otherwise necessary. And then we performed the ASAP-seq assay. And so here I'm going directly to showing you the annotated clusters of the lymph node CD4 positive cells. And you can see, as one might predict, a lot of different T cell populations here, including germinal center TFHs, what are called mantle TFHs, which are CXCR5 low, PD1 low cells, and then various stages of differentiated cells. And we've removed the naive cells, I should note here, because these are memory cells. And then we also pick up some other CD4 expressing cells, such as monocytes and APCs. When we then look at the HIV infected cells, again, now we see many fewer cells, as one might predict. In fact, in this case, we only had about 3,500 or so total cells that were sequenced, and 0.65% of those, or 23 individual cells, came out as having presence of integrated provirus. And what's very clear to see is that those cells are, even though there's few of them in this example, are focused into the TFH populations, as one might predict. And in fact, approximately 40% of the detected infected cells were germinal center TFH or the mantle TFH. And we can see, although this is a few number of cells to do such an analysis, we can see some significant differences in CD71 and ICOS being more highly expressed on the HIV infected cells compared to the entirety of the HIV negative cells. So the final proof of concept experiment that we performed is to look at the most difficult of the examples that one could try, which is looking for HIV infected CD4 T cells from the blood of people living with HIV who are under therapy. So here we expect very low levels of virally infected cells. And so again, we enriched blood memory CD4 T cells by negative selection, did not activate them, and we just simply performed the ASAP-seq assay. Here showing the UMAP projection of the CD4 positive populations from that assay. We can see some inclusion of monocytes and otherwise a variety of different CD4 T cell subpopulations. memory subpopulations. We tested 2 different individuals because we were trying to accumulate more infected cells for deeper analysis. As you can see here, the challenge in detecting these cells in an R-treated individual really comes through. And so in the first individual where we sequenced about 25,000 total CD4 T cells, we only found 5 infected cells, which corresponds to them having a low reservoir size as determined by qPCR analysis performed by Katie Barr. The second individual, we found more infected cells, and this individual also had a higher level of infected cells, predicted to be somewhere between 1 and 2 per 1,000 CD4 T cells. And so you can see here, again, infected cells in a variety of different phenotypes. And we can figure out the particular phenotype of these cells being either late or early differentiated cells. And probably the take-home message here is that the infected cells don't all fall into one bin. They're spread out across different memory phenotypes. But we're still able to find particular markers that seem to be enriched on the infected cells. Here showing CCR5, CD2, PD1, CD162, and CD45 as being selectively higher on the HIV infected cells. So in summary, HIV-infected CD4 T cells can be precisely identified by the presence of integrated proviral DNA using this ASAP-seq procedure that we've defined. We can use this for both in vitro models and ex vivo samples, but the latter has the very large caveat of the cell rarity and the potential to detect cells with defective integrated provirus. The ex vivo sample analysis does not require T cell activation or other manipulation that would bias cellular profiles. So we feel this is giving us a true picture of what the actual cells that are infected are. We can simultaneously identify surface biomarkers and the epigenetic profile of infected and, importantly, the uninfected cells simultaneously, allowing us to control for each one. And deeper analysis, for example, epigenetic profiling of individual genes or of the infected cells themselves, is not possible with our current analysis because of cell numbers. But as we accumulate more data and batch additional individuals' data together, we'll be able to develop this picture. Importantly, we cannot provide the full viral sequence of the integrated viruses through this assay. If the virus is in open chromatin, we can pick up multiple reads across the genome to infer intactness, but this is not an assay that's equivalent to full genome sequencing. And finally, we're in process of adapting this to SIV infection and further to combine this with single cell RNA-seq to allow us to determine transcriptionally active cells in combination with cells that have integrated virus as well. So finally, I'd like to put up my acknowledgement slide, and I'll just really highlight here Vincent Wu, a graduate student in my lab who has done almost everything that I've talked about today, and then our funding sources, some R01s and P01s, most importantly, a supplement provided by the NIH to the Penn CFAR, to Laura Vella, my collaborator on this project, and myself. And I'd like to thank you very much for your attention.
Good afternoon. Let me begin by expressing my gratitude for the opportunity to present at this year's National CFAR meeting. The research I'll be discussing with you today is on some of our recently published work on a signaling mechanism that we identified to be essential for the reactivation of HIV from latency in primary CD4 positive T cells. This research focuses on a host factor known as BEAT-HIV, which is a transcription elongation enzyme that is co-opted by HIV in order to be able to synthesize its viral RNA. Now, the assembly of BEAT-HIV and stimulation of its transcriptional activity in primary T cells are thought to follow a sequence of events that critically depend upon T cell activation signals. So considering that this enzyme is an obligate transcription factor for proviral HIV, this research was aimed at defining T cell signaling pathways that are involved in mediating the biogenesis of transcriptionally active BEAT-HIV. We began by assessing exogenous signals capable of stimulating active P2FB in primary T cells, and observed that whereas co-stimulation through the T cell receptor elicited a maximal effect, protein kinase C agonists such as ingenol, prostratin, and PMA also induced P2FB expression in the absence of co-treatment with a calcium ionophore. And I should point out here that co-treatment with an ionophore, in this case ionomycin, did not further enhance the inducible expression of P2FB. So we set out to examine how these agents, these PKC agonists, signal to stimulate active P2FB. In also assessing the reactivation of latent HIV, we utilized a well-described and an extensively characterized primary Th17 cell model. And using these latently infected Th17 cells, we were able to show that both ingenol and prostratin could sufficiently stimulate active P2FB and also reactivate latent HIV to a similar extent as T cell receptor co-stimulation. However, surprisingly, and in well-controlled experiments, we found that protein kinase C enzymes are not involved in mediating the inducible expression of P2FB or even the reactivation of latent HIV by either ingenol or prostratin in primary T cells. Eventually, we found that these PKC agonists signal through the MAP kinase ACK pathway in order to elicit the post-transcriptional induction of P2FB. Now, activation of the MAP kinase ACK pathway by these PKC agonists is very likely to be mediated by a guanine nucleotide exchange factor known as RAS-GRP, which in T cells has been shown to be required for the initial activation of RAS and the MAP3 kinase RAF. And using both single cell and bulk RNA-seq transcriptome approaches, we were able to demonstrate that of the 4 known isoforms of RAS-GRP, RAS-GRP1 is by far the predominantly expressed isoform in primary T cells. We also conducted Western blotting analysis, which corroborated the RNA-seq results. And by performing high-resolution microscopy, we were able to confirm that RAS-GRP1 can traffic to the plasma membrane of primary T cells in response to brief stimulation with these PKC agonists. So in conclusion, we found that the tight coupling of the biogenesis of P2FB to the reactivation of HIV from latency in primary T cells can be accomplished by treatment with PKC agonists in the absence of intracellular calcium mobilization, and that these agents signal through the alternative activation of the RAS-GRP1, RAS-RAF, MAP kinase ACK pathway in order to be able to stimulate these consecutive processes. Therefore, based on these findings, we believe that selectively targeting this MAP kinase pathway to generate transcriptionally active P2FB may provide an optimal strategy for latency reversal. I thank you for your attention and will be glad to answer questions that you may have.
Hello, everyone. My name is Katie Barr from the Penn CFAR, and it's my pleasure with Luis Montaner to lead a little discussion from this excellent session. So I'm going to start off with a question posed by Dr. Raj Gandhi to Bob Siliciano. Great talk. Are the viral epitopes targeted by the autologous neutralizing antibodies known? Might that information be used to inform development of therapeutic vaccines or antibody combinations?
Yeah, thanks, Raj. Great question. We're trying to determine now precisely where these autologous antibodies bind on the envelope protein. Now, what we do know is that they target variable regions. There's very little cross-neutralization by these autologous neutralizing antibodies on isolates from other individuals. The hope is that we can identify some common feature of the the isolates that are resistant to these autologous neutralizing antibodies. And that would be something that would be important to target for the therapeutic vaccination. We don't know yet whether that will be possible.
So we have a second question by Dr. Coleman to Anne. On thinking of the prior two talks, are you able to examine sequences of your induced viral sequences along with analysis of the reservoir to ask whether you are reactivating the reservoir broadly or only a specific subset of the reservoir? And if I can add to that, if you had any instance of a viral rebound, did that sequence match or not to what was reactivated?
Yeah, thank you for that question, Ron and Luis. That is something that we are very interested in. In the original study where we used the CIAP antagonist, we did not conduct an ART interruption. But we did look at envelope sequences of the virus that reactivated during treatment and compared it to the peak viral load and the pre-ART viral load sequences in our animals. And basically what we were able to conclude from that is that some of those sequences when the treatment is ongoing and the virus is reactivating during the presence of antiretroviral therapy are similar to peak, some are similar to pre-ART, some are clonal, and some are unique. So sort of a conclusion of a bit of a heterogeneous viral rebound. We did not look at, sensitivity to neutralization, for example, which I think could be very interesting. And we do have a new funding now to utilize the Brandon Keel's barcoded virus to kind of look a bit more deeply at a number of different tissues, a number of different cell subsets that may be reactivating the virus. So compare the plasma virus during reactivation to virus found in different cells and tissues. So hopefully we'll generate more data that can help answer that question.
Thank you.
A question for Mike Betts. Your ASAP-seq study was really exciting, and I know it's just preliminarily been used on cells from people living with HIV suppressed on antiretrovirals. And I think it's reassuring that the first markers you find are things classically associated with HIV infection, like CCR5 and PDL or PD1. Are there any novel markers that you've identified from these reservoir cells that you think are particularly interesting as a signal?
Well, thank you for that question. Right now, we're still in the process of accumulating data, and as we get more infected cells from looking at more donors, and we can start to get a little bit better resolution on the other markers. But so right now, almost on a person by person basis, we see slightly different marker patterns come up, but I think it's because we just need to accumulate more data at this point.
So a question for Sue. In your elite controller cohort, you describe how the silent regions could be overrepresented in the elites and certainly in the cure cases. But have you been able to see a relationship between the regular sites and CD8 responses within an individual? It's almost taking it to the other extreme, saying you have individuals that have the silent regions and could sustain the level of the IPDA, but conversely, would you then say that if you look at CD8 pressure or CD8 frequency, that those with more regular regions have greater frequency of CD8 because they would otherwise be still controlling the virus?
No, that's a great question. We have not done a comprehensive immunology study on these patients, but Bruce Walker and Graf group is doing that. Actually, that's exactly what we try to address, whether the CD8 T cell pressure helped clean up the integration site in the accessible chromatin and only left the intact proviruses in the heterochromatin regions still available there in vivo.
Okay, thank you. This is a question for Mr. Carter. I thought your talk was really inspiring. And one of the things you mentioned was asking community members not just to the table, but into the kitchen. And I think as we have this collection of really transformative basic and translational researchers, I wonder your thoughts about bringing community members in as early in the sort of basic and preclinical process of research and how we might do a better job of doing that.
I think by having forums like this is one good way, and also letting it be known that you really want our opinion. Because as I stated there, no matter how much you guys come up with things, your dreams become reality, and your dreams become my reality, which causes me to live. And who better to help you run your product to do that? You have an exclusive audience. So I feel as though you need me as much as I need you. So when you're coming up with the advertisement, the testing of it, why wait until you've already spent millions of dollars to have an idea? And then as soon as you invite me and my colleagues in, it's all wrong. If you're going to use me at the beginning of the planning stages. I may not catch on right away, but I do catch on. So remember that. So when you're speaking to the community, if you're, as RCAB does, we speak as one. No one has an individual voice there. We all come in together. We may not agree at one time, but we do make a consensus together on how to further things. So if you use me, I'm willing to be used. Use me in the advertisements. Use my fellows. Use us. But who better to speak about it than the both of us together?
Thank you.
So Yuri, in regards to latency reversing approaches, as you saw during today's discussion, there is a lot of different strategies that we're trying to reconcile how best to move forward with this particular area. So when you look at what you were able to document with the approach to the TF beta and the SMAC mimetic versus the anti-CD8, what are your thoughts of how you're going to sort of integrate or not these approaches moving forward? How do you see the next steps or the priorities on latency reversal? I know it's a hard question, but it's one that we all kind of wrestled with, and you're deep into it. So I'm just seeking what your opinion is. Are you there? Can you hear me?
Was the question directed at me?
Yes, it was relative to latency reversing approaches. What is the path forward for the work that you're doing with regards to the other latency reversing approaches that we've heard today? How do you balance all these approaches moving forward?
Yeah, thank you. So our work focuses on ways to activate a host factor called BEAT-HIV, which is as I described in my presentation, a transcription elongation factor required for the emergence of HIV from latency. And we've identified the MAP kinase ACK pathway as an essential signaling pathway for targeting BEAT-HIV. So BEAT-HIV acts at the transcription elongation step of proviral HIV transcription. So the other essential step would be activation of transcription initiation. And as Anne has nicely described, it would be possible to do so with a stimulus that activates non-canonical NF-kappa B signaling. So a combination of an agent that would target non-canonical NF-kappa B signaling and an agent such as these PKC agonists, particularly these novel synthetic PKC agonist derivatives, those two, I believe, in combination, might act well to optimally reverse HIV latency.
Thank you. And Bob, you said you had a question?
Just a quick question for Anne. Do you know if the SMAC mimetic's acting directly on CD4 cells, or is the effect indirect?
Well, we presume in vivo that it's acting directly on CD4 T cells, and ex vivo in terms of taking CD4 T cells from HIV-infected ART-suppressed patients, it can induce HIV induction. So there may be additional effects in vivo that we are not, quite capturing from those ex vivo studies, but I think ex vivo we can say yes.
All right, thank you all so much. I do feel that we could go on for several more hours, but unfortunately we have to close here. I want to thank all the speakers. Thank you very much. Have a good rest of the seminar.
Thank you.
Thank you.
Thank
Thanks.
you.